nothing else than the encapsulation response. So this is a very general response. You can implant almost anything, not quite everything, but almost anything, into the hemolymph of these animals and many other insects, of course. And what you get is a response by hemocytes that attach to the surface of the foreign object and start to melanize, which is dark here, and eventually encapsulate it. So this is strength. This is a weak response, this is a strong response, and at the same time we test the same families for their ability to contain a multitude of infections. So these are controlled infections with a cocktail of these strains, and as you can see here on this end, many strains are excluded, so the colonies are actually quite good in eliminating most of the infections of the strains, and here only few are excluded. And what you see here is a negative correlation in the sense that the more stronger your standard or classical, let's say general immune response is, the less able you are to contain a multitude of infections of this parasite, and vice versa. And recall, if you're here and actually are infected by many strains, your chances to get a lot of infected daughter queens is higher, which means they lose a lot of things next year when they start their colony again, their own colony. So you have a compromise either to exclude lots of those here, and at the same time not have a very strong general response. So this is cost in some sense, or trade. The second trade-off turned out to be the case in a completely different context where we didn't expect it in the first place, and this was in the context of transgeneration immune priming. So this is a series of experiments that Ben Satt has done in our lab, where he took some queens, actually infected them with heat-killed bacteria or the controlled sham infections, tested the workers a little bit later, and you can see then, for example, that in this particular case, if you test the workers that come from this mother is primed or not primed, if they are actually primed, the mothers, then actually the workers that come from this mother are better protected against bacteria in the sense they have a strong response against the challenge by bacteria measured here as a zone of inhibition or assay than if they come from mothers that are not challenged. Now you can do the same transgenerational, so this is within generation, so to speak, within the same colony. But you could do essentially a similar thing actually for a next step. And Ben has taken up the challenge, and he has done a challenge, has done a similar experiments where he actually challenged the mother versus non-challenged in a controlled way, and then was looking not so much for the response of the workers as they would be able to defend themselves against bacteria, but actually whether or not they're able to contain this trypanosome parasite. And it turns out, again, you have now a trade-off, not unlike perhaps as you have seen before in this negative correlation, that if you come from one that is challenged and you are immune primed against the bacteria, then you're less able to contain infection by this trypanosome. So again, we have a cost that is not only actually only happening, let's say, inside your cell, so to speak, but it, in this particular case, is primed by the node state. Of course, the trade-off must be within the individual host. So here we have an unusual cost aspect, if you like, that says these different defense performances, regardless of how they come about, have a certain trade-off quality. Using this particular insight, we were then interested whether or not actually by changing the immune status of host individuals, we could have actually provoked a different response for the trypanosome infections. Now, we did this in a standard way. You can see here an example where actually this animal here, which is chilled, so it's put on ice and taken up by here some forceps, and then injected with the antigen in controlled ways with a fine pipette here. You can see this works actually rather well. So what you can do is to challenge these animals, let them recover, of course, and then infect them by the critia, by the trypanosome. And in this particular case, it's not only a single infection, so we're coming back full circle to the beginning, the multiple infection, concurrent infection of different strains in a single host. And this graph shows the effect of immune boosting, or however it's called, on actually the probability that two strains that are experimentally infected coexist or not. So we've seen that before. There's sometimes coexistence among co-infected strains and sometimes not. And in this particular case, the graph reads as follows. Here in the lower line, you have a different combination of strains. So each letter is a strain. Let's say in the first panel here, it's a combination of strain A and B, combination of strain A and C, and so on. And each combination was in controlled ways, in different colonies, and so on, infected into workers that came from, that were controlled workers, so sham infected or sham injected, and in workers that were actually immune boosted, so had a priming. And this is repeated across different all possible combinations of these 4 or 5 strains that we used here. Here on the y-axis you see the proportion of observed cases. Now it reads as follows. So the gray bars, so light gray and dark gray, are the means that actually strain 1 and 2 that was infected, so in this particular case A and B, is existing in the animal. So in this particular case it is strain A that won. This particular case is this 50%, the other 50% roughly is strain 2 that won, and so on. And interesting bit here are the orange lines here, which are the coexistence outcomes. And as you probably can see slightly, actually statistics are okay, but it's not overwhelming significance obviously. Actually what turns out to be the case is that there is a tendency, significant in this case, that if these animals are primed, that it's more likely that these 2 strains that in fact start to coexist. So again, we're full circle back to what I was already showing, that if you do the competition experiment in the media, the first example, versus in vivo, in the animal, it was more likely to coexist in the animal. Now this is both inside the living animal, and depending on the immune status that the animal has, the priming status, it allows the strains to coexist or not to coexist. And again, you can see that by these different challenges here, what you get here is a diversity effect that is maintained, can exchange genes, and then does, of course, also bode evil, so to speak, for the queens because they are more likely to infect. So let me summarize. I think we have a system with very diverse infections. Multiple infections are very common. We see that the parasite plays an important role, obviously. Genetic exchange is quite an engine here. We don't have to forget that it is an epidemic, so it's a stage within which the host finds itself. We find that the strains compete, and we find that the host immune status, so to speak, seems to affect this competition. And of course, this immune defense seems to happen along a line of trade-offs, so the host has somehow to decide whether it's putting more emphasis on containing multitudes of infections or not. And the consequences of that in terms of epidemiology and fitness of the host are actually quite drastic, as you've seen in this example. So, no, I skipped that one. Actually, should be a final slide that says thank you.
For questions?
Yeah.
The colony is founded from a single queen.
That's right.
So a colony is actually relatively genetically inbred.
Yes, they're all full systems.
So do you get local adaptation where that queen infects effectively her colony? There's many generations of the parasite during the life of the colony. It's local adaptation, and then something changes. Well, there's cards are shown.
Yeah, we've done an experiment to that where you infect these with a certain cocktail. And it is filtered out. So after 3 to 4 passages through the individual host, they actually, interestingly enough, they lose the ability to infect other colonies, other families. So they're getting more adapted to this particular one, but they don't get worse as far as you can tell. They don't increase in virulence. It's just that they get more specialized somewhere, which of course is sorting out of types. I mean, it's that kind of thing.
Have you looked at how the gut microbiota composition changes before, during, and after infection?
Yeah, that's what we are about to do now. Yeah, that's an obvious question whether or not actually you find a change in response to the infection itself. No, I mean it was quite unexpected to us to find it. I mean there's some cases now in the literature where people have shown that bacteria, of course, seem to play a role, but it's always surprising to find it in your own system that it works. So we're really excited about it.
So in that last priming experiment, was that prime a general, or was it specific to one of the strains, or was it?
No, this was, in this case it was bacterial priming. So it's usually a heat-killed bacterium, in all cases Arthropacter. but it can do the same with, let's say, a standard agent like LPS or something, works in the same way.
So it looks like you're getting more infection after the priming?
That's right,
Is
yeah.
that consistent, or is that just normal?
No, it's actually very, very consistent that in particular if you then come in with the second parasite, so to speak. So what's happening, they are themselves primed, maybe they have a past infection of their own that they have cleared, they stay primed, or their mother has been primed even and gives it to the offspring. In both cases it seems to work that they get more susceptible to another kind of parasite, which of course shows the internal trade-off. Not surprising, but you can see it here. Maybe surprising, but it's something that is not completely unexpected, but in this particular case it works that way. It seems like opening up space, if you want, the immune space, so that the trypanosomes can invade. Unfortunately, we don't know exactly the mechanism.
So it's the cost emphasis, the trade-off between one or the other was the emphasis. But do you think there's a case where there's an outright vulnerability? So it's only a vulnerability to one parasite or the other, but would there be context where the things are sort of?
No, clearly. I mean, there's normal standard costs, if you like to call it that way. I mean, that's absolutely true. I mean, of course, what you also have is a difference in susceptibilities among families. That's also clear. You can see all of that. You can see normal costs by, I don't know, stressing them and then things go wrong. No, that's all there. But here it's, against this background, you also have apparently an interplay between defenses against different parasites. And this is the natural situation right there. Constantly somehow challenged by bacterial infections, they're constantly challenged by trypanosome infection. In this example, of course, there are also viruses now, as we know. And all these plays together, and you get a very complex mixture of challenges. What I was trying to say is it's good to remember that we should really also go out to the field, so to speak, and find out what these challenges are, and what is the task that the defense system has to do. And here in this particular example, it has to do this trade-off between these two major challenges. Maybe the viruses are already there. Yeah.
Maybe I'm misunderstood, but isn't another explanation not simply a trade-off, but the fact that by priming the bees with bacteria, you're biasing the response down a certain pathway, a particular protection pathway, which is not particularly useful for protection against the trypanosome.
Yeah.
So actually what you've got is an animal that's making a particular response, it's not very good at dealing with trypanosome. It's not necessarily a trade-off, it's simply that.
Well, it's a trade-off in the sense that the individual, so to speak, can decide in inverted commas whether he wants to invest in one function or another. Whether this trade-off in the sense is hardwired, let's say, that is genetically determined, or whether it's plastic is another matter. I mean, we haven't really, here of course, we don't know whether this curve comes about from a hardwired genetic background, or whether it's actually flexible, that the animal can sort of decide what curves it takes, so to speak. So in that sense, it's a trade-off. It's not, let's say, something that is fixed, so to speak, that you cannot do otherwise. So in that sense, it's a trade-off. If that answers the question. Does it? Or?
Well, we could discuss it later.
So, sorry, I think we should move along to keep on schedule. Thanks again, Paul.